Hello friends, welcome to my channel. Today I am telling you about an important topic of microbiology, that is the types of staining. Staining is important for the microbes identification, and it is an important technique for the microbiology. There are three types of staining. First is the simple stain, then the differential stain, and third type is the spatial staining. In simple stain, there are two types, positive staining and negative staining. And differential staining, there are also two types, gram staining and acid fast staining. And spatial staining are of three types, capsule staining, flagella staining, and endospore staining. First is the simple staining. Simple staining means in which we only use the single type of dye for staining purpose. In positive staining, the actual cells are themselves colored and appear in a clear background. In simple staining, we can use the crystal violet dye, green dye. In the differential staining, we can differentiate the different types of microbes. For example, gram positive, gram negative bacteria. For this purpose, we perform the gram staining, and acid fast staining and endospore staining. They are the special type of staining. So different colors uh, impart to the microbes, and on the basis of color the different types of microbes are differentiated. That's why it is called the differential staining. In simple staining, we can it is a single step procedure, and we only use the single type of stain. There are two types of staining, direct staining and positive staining. In this, only the it stain object, and indirect and negative staining, it will stain the background. Means in the positive staining, uh, it will stain the microbes, not the background, but in negative staining it will stain only the background, not the microbes. Negative staining, that is dry film method. In this method, we can use the nigrosine, that is a modification of Indian ink 2% mercurochrome. It can be used, and for this negative staining, we will take a clean glass slide, then place a small drop of nigrosine close to the one end of the slide, and using a sterile loop, a loop full of the liquid culture, means the broth culture, is mixed with the nigrosine dye. With the edge of the second slide, we can make the thin smear, and then we will air dry the slide and observe under the microscope. It is the differential staining type, that is the gram staining. For this, we are using the different types of dyes. First step is the crystal violet. In this, it will act as a primary stain, and it will stain both the gram positive and gram negative bacteria. Then we will use the iodine, and it will form complex with the crystal violet, means the crystal violet iodine complex will be formed, and then result will be the cells still purple, but now more fixed in the gram positive. Then we will stain with the safranin for the 45 seconds. It is a counter stain. Then the gram negative will appear the pink, and gram positive will appear the purple due to the different cell wall compositions. And then in this way, the gram positive and gram negative bacteria will be differentiated. It is the flowchart of different types of the staining, and acid fast staining, that is called the gel Nelson. In this, carbamazepine dye will be used, and heat fix the microbe. Then we will use a decolorizer, that is the acid alcohol and methylene blue, and it will stain the acid fast bacteria, and they will appear under the microscope in red color. Endospore staining, for this purpose malachite green dye will be used, and then heat fix, then the rinse with the water, and then counter stain with the safranin and observe under the microscope. Endospore will be appear green in color. And capsule staining, for this purpose mix the bacteria with the Indian ink and then prepare the slide and observe under the microscope. Capsule will be appeared as a halo. And negative staining, simple staining and gram staining we are already discussed. This is the image of the negative staining. As I earlier told that negative staining only stain the background, not the bacteria, not the microbes. So microbes, in this image microbes are the white in color, and the background is dark in color. It is also the image of negative staining. This is the image of gram staining. Gram positive cocci. Uh, Simple staining only tell about the shape of different types of microbes, means spherical shape, cocci, and the rod shape, comma shape. So it will only tell about the shape of different types of microorganism. Then negative staining, uh, it will stain the background, and gram staining will differentiate between the gram positive and gram negative bacteria. So here is the result of the gram staining, and purple color bacteria are the gram positive bacteria, and they are cocci in shape. That's why they are called the gram positive cocci. And second figure represent the gram negative bacilli. Gram negative means pink color, and bacilli because they are of rod shaped, so that's why they are called the gram negative bacilli. So it is all about the different type of staining. And as we know, the staining is very important to identify different types of microbe based on their shape, based on their color. So it is very important technique for the microbiology.